All right, today let's work on, there was a few things from respiratory system that we haven't discussed yet that we need to. Okay, so we mostly need to talk about the factors that can affect the rate of the diffusion and the rate of the gas exchange. So let me write this out. First of all, we need to know that the exchange of oxygen and carbon dioxide that happens at the alveoli is diffusion, because diffusion is supposed to be the movement of a substance from high concentration to low concentration. So in the lungs, there's a high concentration of oxygen, and then there's a low concentration of oxygen in the blood. So that's how the oxygen knows to flow from the lungs to the blood, is because it's flowing from high to low. Then for the carbon dioxide is in a high concentration in the blood, and then there's a low concentration in the lungs. So that's how the carbon dioxide knows to go from the blood to the lungs. And it's because of that, the difference in the concentrations. Okay, then, like I said, because this is moving from high concentration to low concentration without the help of a protein, that's how we know that it's moving through diffusion. Okay, so now let's talk about the factors affecting the rate of diffusion. So the first one is actually the difference in concentrations. So basically, the bigger the difference in the high concentration to low concentration, then the faster the movement. Because, for example, if you think about like a hill, the bigger the difference between the top of the hill and the bottom of the hill, the faster the movement. So if there's a high concentration of oxygen in the lungs, and then a very, very low concentration of oxygen in the blood, then yeah, definitely oxygen is going to move very rapidly from the lungs to the blood because there's such a big difference in the concentration, just as if there was a large difference between the top of the hill and the bottom of the hill. Okay, now let's take a look at the carbon dioxide. The carbon dioxide doesn't have that much of a difference. It's still going to move from high to low. But yeah, the less of a difference that there is, then the slower the diffusion. So it's going to be 45 in the blood, and then low concentration of 40 in the lungs. So the carbon dioxide is going to move a little bit slower. So this is for oxygen, and then this is for carbon dioxide. It moves a little, it diffuses out of the bloodstream a little slower. Some things that could affect this would be, for example, like exercise. So during exercise, your muscles are going to be consuming and using up the oxygen from the blood. Let me write this out. During exercise, the muscles consume, in other words, use up oxygen from the bloodstream, And so that will actually decrease the blood oxygen concentration. And so what that will do is that will allow for the diffusion to occur even faster out of the lungs and into the blood. Okay, now let's see. The carbon dioxide, meanwhile, we need to know that during exercise, the muscles are going to produce carbon dioxide, and that will increase the blood carbon dioxide concentration. And so that could cause a faster diffusion of the carbon dioxide out of the blood and into the tissues. So that's just the first factor that can affect the rate of the diffusion. Now let's take a look at the second factor that can affect the rate of diffusion, which is actually supposed to be the distance traveled. In other words, the thickness of the membrane. So to understand this, let's actually think for a moment about, like, if we were conducting an experiment, and let's say we had someone standing in a closet, one corner of the closet, and then someone else sprayed perfume in the other corner of the closet, and then we timed how long do we think it's going to take, or how long did it take for the person in the corner to smell the perfume? So let's say that the person in the corner smelled the perfume in 2.7 seconds, which would make sense because the perfume didn't have to travel very far. So of course the person smelled it fast, very quickly. Um, but let's say, well, what happens if let's say that we repeat this experiment, but this time at maybe like a basketball court, or maybe a some other type of gymnasium. We have one person stand at one corner, and then we have the person spray their perfume in the other corner, and we time how long it takes for the person in the corner to smell the perfume. I mean, obviously, because it's a longer distance, it's going to take a lot longer for the person to smell the perfume. So let's say that this time it takes 3,628 seconds, which would make sense. Okay, so the way that this relates to the respiratory system and the gas exchange is because normally what happens is that any kind of capillary is going to be made up of simple squamous epithelia. And simple squamous epithelia, those are very thin, flat cells. So around the alveoli, there are capillaries that have that simple squamous epithelia. And then always between two layers of cells, there's going to be a basement membrane. And then we already know that the alveolar cells are also simple squamous. So the oxygen that's in the alveoli, it doesn't have to travel very far before it gets to the bloodstream. And the carbon dioxide from the blood also doesn't have to travel very far before it gets to the alveoli. So basically, this very, very thin membrane allows for very, very fast gas exchange. So if you're wondering, okay, when would it ever change? How could it possibly be any different? Well, there are some respiratory conditions, well, not necessarily even respiratory, but circulatory conditions where sometimes there can be a backup of fluid in the bloodstream, like in congestive heart failure. It can cause pulmonary edema, and pulmonary edema is where the backup of blood is going to start to push fluids into the tissues of the basement membrane and the alveoli. So let's say that the endothelial cells of the capillary aren't really affected, but then the cells of the basement membrane are pretty swollen, and there's some fluid between the basement membrane and the capillary. And then from there, let's say that after that, maybe the alveolar cells are a tiny bit swollen, but not too bad. Right, and all of this is coming from increased blood pressure at the capillaries around the alveoli, and that's forcing fluid into these tissues, and perhaps maybe even the backup is so bad it's forcing fluid into the alveoli. So we'll have fluid all in here, but then there also could be some fluid in here. And so now the oxygen, which is in the alveoli, is way over here. It has to travel a lot farther now in order to get into the bloodstream, all the way through all of this, and then finally at the capillary. And then the carbon dioxide also has to travel all the way through all of this just to get to the alveoli. The gas exchange will happen. It's still going to have to move from high concentration to low concentration, but it's going to be a lot slower because the gases now have to travel farther. The farther that they have to travel, the slower the movement. Okay, now the third factor that affects the rate of the diffusion is just surface area. Anytime you have increased surface area, you're going to have increased absorption. And so there's going to be an increased amount of gas exchange. And then let me also add that this is thicker. When you have a thick membrane, it results in a much slower gas exchange. Okay. Right now, let's move on to urinary system. So I'm also going to be sending you a video on some of the topics you need to know for urinary system, but we're going to be going over the things that aren't in the video. So first, let's take a look at the structure of the nephron. So we need to know that the nephron is the functional unit of the kidney. Each nephron has two parts to it, a renal corpuscle and then a renal tubule. The renal corpuscle, basically that is supposed to have two different parts to it. It has a Bowman's capsule and then also a glomerulus. Let me add that to the diagram. So this right here is the renal corpuscle, and it has the glomerulus in here. And then all around the glomerulus is supposed to be the Bowman's capsule. And the Bowman's capsule is supposed to be a cup that's going to collect everything that comes out of the glomerulus. And right, the glomerulus, the key word for that is that it's a capillary. Yeah, definitely need to know that it's supposed to be a capillary. Okay, now all the rest of the nephron is supposed to be the renal tubule. So let me draw that out. And then all the rest of this is the renal tubule. And I might have to move it a little bit. Yeah, there we go. Right, all the rest of this is the renal tubule. Renal tubule. The renal corpuscles in orange. So the renal tubule has several parts to it. It's supposed to have a proximal convoluted tubule. So the proximal convoluted tubule is all this part in here. Then there's the loop of Henle. The loop of Henle is this structure right here. And then we have the distal convoluted tubule. And then after the distal convoluted tubule is the collecting duct. A lot of places don't really consider the collecting duct a true part of the nephron. So just keep that in mind. All right, now let's take a look at more specifically the histology of the glomerulus and the Bowman's capsule and the rest of the renal tubule. Okay, so we need to know that each renal corpuscle is supposed to have a glomerulus and then a Bowman's capsule. So as I mentioned, the key word for glomerulus is the word capillary. It's supposed to be a network of blood capillaries that surround the Bowman's capsule. We need to know that the glomerulus is fenestrated, meaning that there are gaps in between some of the cells. So this diagram in here, the endothelial glomerular cells are in red. And you can see that there are the fenestrations, the gaps between the cells. Leading into the glomerulus is the afferent arteriole. So the afferent arteriole is going in, and then the efferent arteriole is supposed to be E for exiting. Let me also add that. Afferent is A for arriving, and then efferent is supposed to be E for exiting the glomerulus. We do need to know that the efferent arteriole has a smaller diameter than the afferent arteriole. Okay, right, the Bowman's capsule is supposed to be a cup-like structure. It has an outer layer of epithelial cells that form a parietal layer, and then it has a visceral layer of podocytes. So the green cells that I drew, excuse me, those would technically be podocytes of the Bowman's capsule in green. And in between two layers of cells, there's always a basement membrane, which I think I mentioned when we were just talking about the respiratory system and the alveoli. There's also a basement membrane there. Okay, and then right, basically we need to know that the podocytes of the Bowman's capsule, they have processes that are called pedicles that wrap around the capillaries. There are teeny tiny gaps between the pedicles called filtration slits. that are going to allow the passage of tiny molecules and ions. Right. And so this is going to allow only the smallest of the smallest of the smallest of particles to be able to pass through. Okay. Now, let's take a look at the histology of the rest of the nephron, and then we'll look at the process of filtration and everything else. Right. So a lot of the nephron is going to be made up of simple cuboidal epithelia. The proximal convoluted tubule has cuboidal cells with lots of microvilli, and that's going to help with reabsorption. And reabsorption means putting things back into the nephron. And then secretion is talking about taking things out of the blood and putting them into the nephron. The microvilli are pretty unique only to the proximal convoluted tubule. So you're not going to really see you're not going to really see microvilli in a lot of the other parts of the nephron. Okay, then after that, there's going to be the loop of Henle. So the loop of Henle has a descending limb that helps reabsorb water and an ascending limb that helps reabsorb ions like sodium. We need to know that the bottom of that loop of the loop of Henle is simple squamous epithelia, which is very unique. The only other place where that's really found is in the glomerulus. And then the thick ascending limb is going to be simple cuboidal. So it goes back to being simple cuboidal again. Okay, then after that, we have the distal convoluted tubule. Also has lots of cuboidal cells. It's also going to help with reabsorption and secretion, but just not quite as much as the proximal convoluted tubule. Proximal convoluted tubule has tons of reabsorption and putting things back into the bloodstream. Now let's look at the collecting duct. The collecting tubules are lined with simple cuboidal epithelia, but they're going to form columnar cells as they get closer to the collecting duct. Right, we need to know that the collecting duct actually has two different types of cells. There's going to be principal cells and then intercalated cells. The principal cells help reabsorb sodium and water under the influence of ADH and aldosterone. And the intercalated cells, those will be important in the acid-base balance and also helping to reabsorb sodium. Okay. And then we also need to know that in the collecting tubule, there really aren't very many microvilli, but there are a lot of microvilli in the collecting duct. So there was a lot of microvilli in the proximal convoluted tubule, and now back in the collecting duct, there's a lot. Okay, now I think that's it for the histology part. So now let's get ready to work on the formation of urine. Okay, so right, the formation of urine begins with glomerular filtration. This non-specific filtration is driven by hydrostatic pressure of blood. So important to know that. And what they mean by hydrostatic pressure really is just blood pressure. Basically, it's just this force out. Basically, the push of the blood against the wall of the capillary. Right. We do need to remember that this pressure is supposed to be higher in the glomerulus than in other capillaries because of that efferent arteriole being smaller than the afferent. So this is the afferent arteriole here, wide, and then the efferent arteriole, which is very narrow. So because of this smaller opening, if you think about it as blood tries to flow through from the afferent arteriole to the efferent, I mean, there's going to be this backup of blood. Not all the blood is going to be able to flow through there all at the same time. So as there starts to be this backup of blood, it's going to start to push against the walls of the glomerulus. And so this push, this blood pressure, is going to start to force small solutes and water out of the bloodstream and into the Bowman's capsule. So let's take a look now. Again, let me relabel this that this is the red cells are the endothelial cells of the glomerulus, right? The blue cells are the basement membrane, and then the green cells are the Bowman's capsule podocytes. Okay, now, right, as that blood pressure is increasing and it's forcing solutes to move through, let's say, okay, one thing that we need to know is that the fenestrations in the glomerulus, they are too small for red blood cells. Red blood cells are not supposed to be able to pass through the fenestrations of the glomerulus. So you can even see that in the diagram that the red blood cell is huge and it can't fit through those fenestrations. Right? The fenestrations in the glomerulus prevent the passage of red blood cells. So that's why you're not supposed to be able to find red blood cells in urine. Is because the fenestrations in the glomerulus are supposed to be too small for that. Okay, then there are some gaps in the cells of the basement membrane, but they're supposed to be too small for plasma proteins like albumin. So the green substances that I drew, those molecules, that would be like the albumin. So you could see how maybe that could fit through the fenestrations of the glomerulus, but then it can't fit through the slits in the basement membrane. So that's also why you're not supposed to find huge proteins in the urine either. Amino acids are one thing, but not large proteins. Okay, then let's see the filtration slits in the podocytes. Those are too small for really anything. Only the smallest of the smallest of the smallest of particles um, can pass. Okay. So that's what happens at the glomerulus is the blood pressure is pushing stuff out into the Bowman's capsule, and that process of them pushing it out, that's going to be the filtration. Now let's take a look at the other parts of the nephron and what they do. First, we need to be familiar with the term secretion and reabsorption and what those are supposed to mean. Okay, so we need to know that secretion means taking substances out of the blood and putting them in the urine. And then reabsorption is supposed to be putting things back into the blood. It's supposed to be putting things back into the bloodstream. Okay, now let's take a look at the functions of each of the parts of the nephron. Okay, so for the proximal convoluted tubule, we need to know that the proximal convoluted tubule is supposed to be the main site of reabsorption. So almost everything that was taken out of the glomerulus is going to immediately be put back into the bloodstream. It's going to be put back into the bloodstream. Then we also need to know, okay, well, what are some examples of things that are going to be put back into the bloodstream? Okay, so that would be things like glucose, amino acids, vitamins, sodium, all those ions, bicarbonate, phosphate, water, and urea. Okay, and then the proximal convoluted tubule will put some things back into the blood, like hydrogen, creatinine, ammonium, drugs, and toxins. Okay, now let's take a look at the loop of Henle. So we need to know that for the descending loop of Henle, that's going to put water back into the blood, and it takes urea out of the blood and puts it in the urine. The ascending loop of Henle is mostly going to be taking a whole bunch of things out of the blood or putting things back into the blood. The sodium, chlorine, all those ions, it's putting them back into the bloodstream. Okay, now for the distal convoluted tubule. For the distal convoluted tubule, that's going to be putting lots of ions back into the blood. The chlorine, calcium, water. Okay, and then how much sodium it puts back into the blood will depend on the level of aldosterone. Okay, and then also it's going to be putting potassium and hydrogen. It's taking that out of the blood and putting it in the urine. Now for the collecting duct, we need to know that that's going to be putting urea, bicarbonate, and water back into the blood. And how much water and sodium are put back into the blood will depend on how much aldosterone and ADH. And then, right, it's going to be putting, it's taking potassium and hydrogen out of the blood and into the urine. Okay. Now, let's see. Let's take a look at homeostasis. Okay, so we need to know how the kidneys play a role in homeostasis. So, right, first of all, right, when osmoreceptors detect an increase in blood osmolality, or when baroreceptors detect a decrease in blood pressure, then the pituitary gland is going to make or release the hormone antidiuretic hormone. Basically, a diuretic is going to make you lose water and pee it out. So, an antidiuretic hormone is going to do the opposite. An antidiuretic hormone is going to help you conserve water and retain water. Right. And so, right, less water is going to be removed in the urine. And so basically water is going to be put back into the blood. And so as more water is put back into the blood, that will help increase blood volume and therefore help increase blood pressure. Okay, and then let's take a look at the kidneys. The way that they're going to also regulate blood pressure um, is through the renin-angiotensin-aldosterone system. Okay, so the renin-angiotensin-aldosterone system, basically what happens is that the kidneys wrapped around the afferent arteriole of the glomerulus, there are special cells called juxtaglomerular cells. Okay, and so the juxtaglomerular cells, what they are going to do is they are supposed to detect a drop in blood pressure. Okay, so, and what they do is when they detect that drop in blood pressure, they will secrete the enzyme renin. Okay, and so we need to know that the renin is going to convert angiotensinogen into angiotensin 1. So one hint is that anything that ends in ogen is inactive. And so the renin activates the angiotensinogen, and that will convert it into angiotensin 1. Then the angiotensin 1 is going to travel to the lungs, actually, and then it's going to be converted into angiotensin 2, right? There's an enzyme in the lungs um, called angiotensin converting enzyme. And the angiotensin converting enzyme will convert the angiotensin 1 into angiotensin 2. Then the angiotensin 2 is the one that has the really powerful effects. It's going to directly stimulate vasoconstriction, so the tightening and the narrowing of the blood vessels, and that definitely will increase blood pressure. And it also will tell the adrenal glands to release aldosterone. And then the aldosterone is going to help reabsorb sodium. And then the more sodium that goes back into the blood, then water will follow. Water always follows sodium. So if sodium is going back into the blood, so will water. And so then that's going to increase blood volume, which is then going to increase blood pressure. Okay. Okay, now let's take a look at the osmoregulation. Okay, so the kidneys are actually the main organ involved in regulating the water balance. Right, so dehydration or excessive salt intake will raise the blood osmolality. So in other words, what that means is that it's going to cause the blood to become more concentrated. So when the osmoreceptors in the hypothalamus detect an increase in the blood osmolality, then the hypothalamus is going to tell the pituitary gland to release anti-diuretic hormone. Okay, so and then anti-diuretic hormone, we've talked about it a little bit, but specifically it's going to go to places like the distal convoluted tubule and the collecting duct and tell them to allow more water to flow out of the collecting duct and back into the blood. It's going to be water is going to be returned to the blood. And then when that happens, then that's going to decrease the blood osmolality. So now it's going to become more dilute. Okay, then aldosterone also helps with osmoregulation. Right, when blood pressure is low, this hormone is secreted by the adrenal cortex. And then aldosterone is supposed to help increase sodium reabsorption. And like I said, wherever sodium goes, water will follow. So if more sodium is going back into the blood, then so will water. And so if more water is going back into the blood, then that can help increase the blood volume, which will then help increase the blood pressure. Okay, now let's take a look at the acid-base balance. So we need to know that the kidneys and the respiratory system together will help control pH. So respiratory and then urinary. This partially relates to what we talked about with hyperventilation and hypoventilation and the bicarbonate. So let's rewrite this. So after cellular respiration, we're going to have water and carbon dioxide combining in a reversible reaction to make carbonic acid. And then the carbonic acid can break down into hydrogen ions and bicarbonate, HCO3 negative. And then let me also relabel, let me move it down actually a tiny bit. That this is this is carbonic acid. And this is bicarbonate. Okay, so what happens is that during hypoventilation, we should remember that the respiratory rate is too slow. And so that's going to cause an increase, a buildup in the carbon dioxide levels of the blood. Okay, and then what that's going to do is that's going to cause, right, these double arrows indicate that the reaction is reversible and that it can increase, or yeah, it's going to cause the reaction to shift. It's going to cause the reaction to shift to the right. And so that's going to increase the concentration of hydrogen ions, which is going to make the blood more acidic. So we need to know, okay, how can the kidneys fix this? What can the kidneys do to fix the increase in the hydrogen ion concentration and the blood becoming more acidic, too acidic? So what the kidneys can do is, first of all, they can secrete and get rid of hydrogen ions. So that means that they take it out of the blood and put it in the urine. Take out of blood and put into urine. Take out of blood and put into the urine. Okay, then another way that they can fix the rise in the, or I'm sorry, not the rise, the decrease in the pH in the blood becoming more acidic is that they can reabsorb bicarbonate. So put more bicarbonate back into the blood. Because the more bicarbonate that they put back into the blood, then that can combine with the hydrogen ions and cancel them out, neutralize them. Okay, so that's what they would do. And then also the last response that the kidneys can have is they can, the intercalated cells of the collecting duct, they can make bicarbonate. They have the ability to make the bicarbonate and put that into the bloodstream. So that's also important to know, is that they have that ability to make the bicarbonate and put that in the blood. Okay. Now let's take a look at, well, what happens during hyperventilation. So as a reminder, during hyperventilation, the person's respiratory rate is too high. And so that means that they're actually breathing out too much carbon dioxide. And as they breathe out too much carbon dioxide, right, the carbon dioxide levels over here are going to go down. But let me pick a different. I guess I'll have to pick this. The carbon dioxide levels are going to go down. And that's actually going to cause the reaction to shift to the left. And as the reaction shifts to the left, that will actually mean that the concentration of the hydrogen ions is going to go down. And as the concentration of hydrogen ions go down, that means that the blood is going to become less acidic. And in other words, more alkaline or more basic. And this was supposed to be cause respiratory alkalosis. So what the kidneys can do to fix this is that they can, they can do this several ways. First of all, one thing that they can do is they can, let's see, they can reabsorb hydrogen ions. So in other words, put back into blood, put back into the blood. And they can also take out bicarbonate. They can take out of blood and put into the urine. Yep. Take that out of the blood and then put that into the urine. Okay, now I think that's it for the acid-base balance. Now let's take a look at removal of nitrogenous waste. So let's first start with proteins. So when we eat proteins, the proteins get broken down into amino acids. But then the amino acids are going to get broken down into ammonia. And then the ammonia can be broken down into urea. Ammonia should be NH3. And then urea is NH2. So the proteins to amino acids, that's done by the digestive system. But then the amino acids to the urea, that's actually done by the liver. So the liver is going to help convert the amino acids into urea. Okay, now we need to know that for the urea, most of the urea is actually going to be put back into the blood, and very little of it is going to go into the urine. So like about, let's say 80% goes back into the blood, is reabsorbed, and only 20% is found in the urine. Okay, now the other types of nitrogenous waste come from, for example, nucleic acids. Nucleic acids are supposed to be things like DNA and RNA. And the nucleic acids are going to get broken down into nucleotides. There's actually two different types of nucleotides. There's purines and then pyrimidines. You need to know this for the biology section anyway about the different types of nucleotides. The way to remember which bases are purines versus pyrimidines is you can use the phrase G, cat, puppy, puppy, like this. So that means that guanine, G, is a purine. C, cytosine, is a pyrimidine. A, adenine, is a purine. And the T, thymine, is a pyrimidine. So we're saying that the purines, the adenine and guanine, are going to get turned into uric acid. So the purines would be the guanine and the adenine. They get turned into uric acid. And so the uric acid, let's say 90% of that is put back into the blood. That means that only 10% of that is found in the urine. So again, most of it is put back into the blood, and only 10% of it is in the urine. Okay, but still urea is the most abundant nitrogenous waste. Okay, now let's take a look at the last one, which is creatinine. So creatinine comes from creatine phosphate. Creatine phosphate is found inside of muscles and is supposed to help store extra energy. And so the creatine phosphate can be broken down into the creatinine, but we need to know that all of the creatinine is going to go in the urine. All of it. None of it is going to go back into the blood. It's taken out at the proximal convoluted tubule and it's never put back into the blood. Okay,